So next we talk about gene ontology. If we have differentially expressed genes and you have also run clustering, so you can imagine you run a k-means clustering on the differentially expressed genes, you can cluster them into different groups. At the end, you don't want to just report the whole list of genes to the user. Or when you have to write a paper, you don't give them, oh, cluster 1 is about this 50 gene names, and cluster 2 is about those 200 gene names. It's impossible to report that in a paper, right? Yeah, so you want to use some text way to say genes that are differentially expressed in this case is really relevant to metabolism or B-cell activation. You want to describe it in a biological, meaningful way. So you want to say that whether things are enriched in some pathways, some functions, or proteins that are related to a complex, or these are all the genes in the nucleus, or belong to some class, say chromatin factors, or things like that. Yeah, so the Gene Ontology Consortium was started by Michael Ashburner in 1998. So he is a very established biologist, and he said, you know, we really need to have a coherent way to annotate each gene. If we can do this in human, then you can extend to many other model organisms. For example, traditionally, whoever discovered the function of a gene can name their gene their way. This is quite crazy in the fly community. So genes can be named with very strange names, right? If you are working on that pathway or that function, you understand the gene, but most of people, if you give them a list of 200 genes, they have no way of understanding what's going on. In addition, people have found, for example, this gene in human is the same or similar gene in mouse, which is called a different name, and it's called a crazy name in fly, even though they might have some similar names. So Gene Ontology Consortium is trying to consolidate that so that you can create a more coherent annotation. Yeah, so why do we care? We want to effectively communicate a biological knowledge, right? We also want to organize and summarize annotations in a structured way. Especially, we want the computer, an algorithm to understand the gene names or the gene meaning so that they can do calculations on this and annotations on those clusters. So you can see gene ontology is created by biologists for the biologists. It's a very simple way. So every gene is annotated with 3 categories. The first one is molecular function. It describes the gene's job or abilities. For example, a gene could be a transporter, move molecules or chemicals in and out of the cell. It could be a transcription factor, which means they bind to the DNA and regulate nearby gene expression. Right? So that's molecular function. The second is biological process, which annotates the events or the pathway this particular gene participates in. For example, some genes might be involved in cell differentiation, maturation, or development process. The third is a cellular component which describes the location of this protein once it's translated into a protein. For example, its subcellular structure. Is it on the cell surface? Is it in the nucleus? Is it in the mitochondria? Whether they form some macromolecular complexes, you know, whether, say, 5 proteins form a big protein complex. So you want to annotate that. So every gene is given those 3 terms. And once you have the 3, okay, you have some idea what this gene does, even though the name could be crazy. And if you look at the gene ontology, it's actually a tree. This of course, is a slide. It's not going to click, but if you can imagine, anytime you click on this, it will open up. You know, this one you click, it open up. So it's like a tree. Every branch can have more branches deeper. So the very top, you have all the goal terms, which is 183,000. But within there are 3 things. You know, there's biological process, there's a cellular component, and molecular function. And you notice that these 3 will add up to more than 183. So, because each is about 110. It should add up to 300K. So it means there are some terms, the same term are being used in multiple locations in the branch, but they want to make sure that there's no circles. So you can imagine this is like the root, and then all these other nodes are like branches in the tree, but you can't have a branch that go back to the earlier branch. And if you look at this relationship, so this is the relationship. Most of the time, you see either a I or a P. The I is a relationship that means is a, and the P is a relationship that's a part of. For example, the 5' UTR is part of a transcript. So if you have an RNA, it has a 5', it has a 3', it has, like, or in the middle, whatever. UTR is the untranslated region. That's just the RNA that will not be made into a protein. Yeah, so it's only part of a transcript. But mRNA is a transcript. It's a type of transcript. It's a whole, like, transcript, but a different type. But you can imagine every time we click into this node, we can have an even more detailed, more specific annotation of some function or some biological process. So at the very deep root of the tree is a very general term. So you can imagine for some genes, we only know there's a new gene, we don't know what it is, we just make some guesses. We only know a little bit. But as people do more research on it, they will know this better and better. Then they will get assigned a more and more specific term on the gene ontology tree. Okay? But yeah, so the same term could be annotated at multiple branches, but at the end we have a directed acyclic graph. So there's a relationship. The branch definitely is lower, or is like thinner than the parent in some sense. So you know the direction. This one is a parent node. This is like a child node. And you can't have a child node that have another node which is a parent for another. So you can't have cycles in this tree. And so every gene is annotated as one term in the biological process with one term in the cellular component and another term in the molecular function. So for example, if a gene is annotated as having this particular Go term, like 46739, which is viral spread within host, then it will also, by definition, it will also be given also this term and also this term. So anytime you have a gene ontology, like a gene annotated with a particular gene ontology term on the tree, anything like towards the root of the tree, any node towards the root will also be assigned to this gene. Okay? So as I mentioned, a new gene, we don't know it very well. You might assign it to some gene go terms that are closer to the root. But the gene that's understood very, very well will be annotated very, very specific terms. And so these gene ontology annotations are still in updates all the time. As people know more and more about the gene, they get annotated with more and more detailed annotations. Okay, yeah, question? Yes. Every gene is annotated to have one biological process, one cellular component, and one molecular function. But if, as I mentioned, if a gene is annotated as some node, everything towards the root, depending on which one, towards the root, every node for this branch is also assigned to this gene. Those probably won't happen, except that if you click this open, you might see a term that belongs to that one. It will be a different term. Yeah, so that's why some terms might appear in multiple places. Okay? Yeah, so how do we use it to really annotate some, say, gene expression clusters or differentially expressed genes? So you can imagine the situation. Supposedly, on the whole genome, we have 20,000 genes for this microarray, and the 200 genes are upregulated. But if you look at a specific gene ontology term, out of this whole genome, only 100 genes are annotated as this Go term. But out of the 200 differential or upregulated genes, 80 contain this Go term. You can imagine this is a highly, genes assigned to that gene ontology term is highly enriched for whatever this Go term is, that pathway or that function, or right? So how do you do this type of analysis? You can imagine this is like a 2x2 table. If we look at the total is 20,000 genes, right? Sorry, let me move this. Yeah, so in the genome in total, there are 20,000 genes. 100 has this gene ontology term. The remaining ones don't have the term. And if we look at the genes that are upregulated, there are total 200 genes, 80 annotated as having this gene ontology, and 120 are upregulated but not annotated to have this Go term. This is what we observe. Then we need to calculate what we expect. How do you calculate, for example, this number? How many do you expect to have this Go term but are not upregulated? How do you calculate this? Yeah. Yeah, it's the 100 times. So basically, you look at all the genes with this gene ontology. You look at all the genes that are not upregulated. You multiply this, divided by your total. That's your expected. Okay, so you can calculate this for each of the 4 terms. And then you can use chi-square test, or you can use Fisher's exact test. Chi-square test basically is just you observe this much and you expect this much for each of the 4 items on this table. So this 2x2 is really the middle 4 cells. Having this Go term upregulated, not having this Go term upregulated. Go term not upregulated, and not this Go term not upregulated. You calculate these 4 scenarios. What's the observed minus expected, and divided by the expected, you add them up, you check your chi-square table. This will be a ridiculously significant p-value, a tiny, tiny p-value in this case, right? And then you say, these whatever experimental condition is, the genes that are upregulated are highly enriched in this particular pathway or biological process. Do we need to worry about multiple hypothesis testing? Right, which categories do you test? If you just have genes in an expression cluster, you have like 50 genes, or if these 200 or whatever, 300 genes are upregulated, you don't know a priori what gene ontology category to test. So the result is you test all 183,000 of them. In fact, the trick is in this course, every time I ask you, do we need to worry about multiple hypothesis testing? Most likely the answer is yes. Right? Every time you have to do every Go term you test. Right? So for example, as we mentioned, if a gene is assigned to a branch Go term, it will also inherit the parent term. And so when this gene is being tested for another Go term, that's the parent, that gene will also be considered. Right? So you can see we have to test all the possible Go terms to see this enrichment. And so there are, for a while, hundreds of tools developed. And by this time, a few are fairly popular. There's Amigo, there's Gorilla. So they very often have a Go, like a geo. There's Grade. And in this homework, we're going to ask you to try a tool called David. You put a list of genes in here, and you can choose the genome-wide, all the genes as your background, or you can feed it another bigger set of genes. And then it will automatically help you calculate what genes or pathways or biological functions are enriched. Okay? And the nice thing about the David is the ones that are kind of similar or kind of on the same branch, it will try to consolidate them together. It's kind of more user-friendly to see. Okay? Questions?